Hi everyone, how's it going tonight? All right, are we ready to learn some science from our country's finest? My name is Julia Ray. I'm the manager of adult programs here. And before we continue, please silence your cell phones and also take note of our handy dandy auditorium doors in case you want to go out to dinner afterwards. You can go out those doors, take you through the green, right downtown, and the parking lot is that way. The library will be closed at the end of this event. So tonight, what are DNA and RNA? Embark with Briana, Kerry, and Alexandra on a journey through the world of nucleic acids, the molecules that encode life. We will learn what they are, how they are processed, and what consequences errors in the code may have on human health, including cancer and Alzheimer's disease. Without further ado, please join me in welcoming our first presenter to the stage, Kerry. Thank you, Kerry.
Hi, everyone. My name is Kerry Larkin, and I'm excited to kick off our presentation this evening entitled Mastering the Code of Life, DNA and RNA Demystified. So before we jump in to the science, I want to take a minute to introduce the three of us who will be speaking today. So as I mentioned, my name is Kerry. I am a second-year graduate student in the Department of Cell Biology at the Yale School of Medicine. My research interests include cancer genome integrity and cell death, and outside of the lab, I like to play the trumpet. Our second speaker is Dr. Alexandra Bregfort. Alexandra, if you want to wave. She is a postdoctoral fellow in the Department of Molecular Biophysics and Biochemistry at Yale. She does research on RNA splicing, and a fun fact about her is she enjoys surfing and dancing the tango. And then our third speaker tonight is Briana Wyman. Briana, if you want to wave. She is a postgrad associate in the Department of Psychiatry at Yale, and she does research on genomics and Alzheimer's disease. And a fun fact about Briana is she likes to hike. Before we get into the DNA and RNA that we're going to be talking about this evening, we need to talk about the fact that our bodies are composed of these subunits called cells. And cells are responsible for carrying out all the functions in our bodies, everything from telling our hearts to beat to sending electrical signals in the brain. But with all of these jobs that cells have to do, how do cells know what to do when? And that knowledge comes down to these three major types of molecules that are inside every type of cell: DNA, RNA, and proteins. And to break down a little bit what these molecules are, DNA we can sort of think of this foundational code, sort of like a computer code. It contains all of the information about us, all our genetic information in our genes that we're going to get into an example of in a little bit. This information has to get copied in the cell and read by the cell, which is sort of like printing out this information from the computer onto paper in sort of a new form. And then once it's in this new form of RNA, this information can be read by the cell and made into proteins. And proteins are really these constructional tools that carry out all of the jobs that the cell has to do. And the focus of our talk today is going to be on these first two types of molecules, DNA and RNA. And these together are called nucleic acids. And they really are sort of the foundational code for life. They include all of the information that makes us who we are. So in between different individuals, there's a lot of variation or differences between each groups of nucleic acids. And a lot of this variation is healthy and makes us who we are. So an example is that each of us has a gene that encodes what color eye we have. So some individuals have a gene that encodes for blue eyes. This information is written in our DNA in this code, and the information gets copied into RNA in the cell, and this information tells the proteins of the cells to generate a blue color in our eyes. And in contrast, some individuals will instead have a gene that encodes for brown eye color, and then the proteins will make brown eyes in this individual. So a lot of the differences in our DNA and RNA is a good thing and is natural kind of out in the world. However, there are certain genes and parts of our DNA and RNA that we do need to be functioning properly and in a specific way to keep us healthy as individuals and to prevent certain kinds of diseases. So if we experience a change in our DNA or a change in one of our genes, this mistake or error might get copied into RNA, and then this leads to our proteins being made either improperly or in the wrong amount, and this can lead to different diseases. In contrast, we may have a part of our DNA that is completely the same as a healthy version, but the cell may make an error in generating this RNA that needs to copy this information, and then we still land at the same place of these proper proteins being made improperly, leading to disease. And when I say disease in this context, some examples of this refer to development of cancer or Alzheimer's disease, which are two examples that we're going to go into today. And these are particularly challenging diseases to treat because unlike, something outside like a pathogen that's going to infect us and cause a cold that we want to eradicate from the body, the foundational causes of these are the same information that make us who we are. So overall, the goal of our 3 talks today is to address where does some of this variation and differences in our nucleic acids, or DNA and RNA, actually come from, and what does this change mean for risk of disease and development of disease. So we're going to attack this from kind of 3 angles. The first, I'm going to talk about how certain environmental exposures, like the sun, for example, can damage our DNA, leading to changes that can increase risk for cancer development. Alexandra, secondly, is going to be talking about splicing, which is an important process revolving around the generation of RNA, that when this goes wrong inside of our cells, can also affect our risk for certain diseases. And then finally, Briana is going to talk about how understanding of differences in our DNA can help us treat Alzheimer's disease better, and how, in general, knowledge of variation in our nucleic acids can help us understand how to treat disease. Okay, so to get into the first part of our talk, I'm going to be talking about the impact of sun exposure on our DNA. So as we're getting into kind of the late spring and early summer and have all this beautiful weather happening lately, I'm sure many of us are familiar with a type of infographic like this, warning us how to protect our skin when we're out in the sun for extended periods of time. So this image here is depicting the guidance from the CDC, the Center for Disease Control and Prevention, advising us, seek out shade, cover up with clothing or sunglasses or a hat, or wear sunscreen when we're out in the sun for too long. And this is because it's been shown that too much sun exposure is associated with a higher risk for cancer, and specifically skin cancer. But I want to break down in my talk today what this actually means and how we get to this outcome from too much sun exposure. And when I say cancer and increased cancer risk, what I'm referring to is uncontrolled cell growth. And the way we can think of cells kind of becoming cancerous is sort of thinking about the way a car works and obeys stop signs. So there are times when we want, the car to be driving down the road, but in order for a car to work safely, it needs to adhere to these stop signs. But if we get to a place where either keeping our foot on the gas for too long or we lose the brakes, we get to this place of the car blowing through these stop signs and continuing to go. And this is sort of similar to what happens when healthy cells become cancerous cells. So as cells are growing, they will divide and copy themselves to create more of them. And this is important sometimes in cases like when our bodies are growing larger, we need more cells to fill them out. We do need our cells to divide and replicate and copy themselves. But cells importantly make certain proteins that act as stop signs and say, okay, we've copied enough, we have enough cells, stop growing. And when cells become cancerous, they can lose their adherence to these stop signs in a couple of ways. They might have errors in making the proteins that make the actual stop signs, so the signal isn't here anymore, or they may make other proteins that tell the cell to keep growing despite this stop sign. But regardless, we reach this place where we have too many cells that are continuing to grow and survive without dying, and this is the point where we reach cancer and we reach developing tumors from these cells. But how does sun exposure actually lead to this point of cancer? And the answer lies in the UV radiation that's produced from the sun. So in addition to the visible light that we see out all around us from the sun every day, the sun is also producing these invisible waves of light called UV radiation that are broken down into 3 types of groups: UVA, UVB, UVC. And these are problematic because even though we can't see them, they have higher energy than the visible light we see all around. And UVC light, this type that is the highest energy over here, isn't really a problem for the rest of the talk that we're going to be discussing because it gets caught in the ozone layer in the atmosphere and never reaches us here down on Earth. However, UVB and UVA radiation both have the ability to penetrate through the atmosphere and reach us, and specifically reach our skin, as our skin is most often exposed to the sun kind of every day. And this is a problem when these higher energy UV radiation waves reach our skin because, as I mentioned, our skin contains cells, and our cells contain DNA, this important coding molecule. And because these waves are higher energy, the UV radiation can damage DNA. But how does it actually do this? And in order to get into this question, we need to first break down a little bit more the structure of DNA and how the cell deals with DNA damage. So I've been depicting DNA up to this point as this double-stranded molecule. You can see these 2 strands kind of overlapping with each other. Broken down further, each of these strands is made up of these building blocks called nucleotides. And if we zoom in on the structure of DNA, that looks something like this. And there are two pieces to this that are important to know. The first is that each strand of DNA, the top and the bottom here, is a sequence of nucleotides in a particular order. And you can see it's sort of a subunit of these three shapes all within this square. And while parts of each nucleotide are the same, what I hope you can appreciate is that this part, which is called the base, can be one of four things, kind of making each nucleotide unique. And each of these strands has a specific sequence of nucleotides, and importantly, each base of a nucleotide has one specific pairing partner across the strand, which allows each of the strands of DNA to come together and actually form this structure that it needs to have. And the order of nucleotides on each of these sequences is important, sort of in the way that the spelling of words is important for us to read them and understand them. The order of nucleotides needs to be correct for the cell to understand the message and relay it to the rest of the cell properly. So the order of paired nucleotides overall is important for the cells to work as they're supposed to. Now, different factors can damage DNA or lead to changes of the sequence in a variety of ways. One of the ways that I'm going to go through here is as the cell is constructing DNA, it may make an error in reading which base and which nucleotide is supposed to pair from one strand to the other. As you see, the cell has made a mistake here in which nucleotide it was supposed to pair. But this is okay because the cell has mechanisms in place called DNA repair proteins, and these work in a variety of ways. But in this case, the repair proteins can recognize that the cell has made a mistake, and they can come in and cut out that nucleotide and replace it with the proper one. So under kind of healthy, normal conditions, even if the cell is experiencing a little bit of DNA damage, that's okay because its ability to repair the damage properly balances the amount of damage that it will experience. But now to get back to the sun, what happens now when too much UV radiation hits cells? And the first thing that happens is that on the way to the DNA, the UV radiation is going to experience this protein called melanin. And what melanin is it's a protein in our cells that is responsible for producing the pigment or the color of our skin and hair. So individuals who have darker skin or darker hair are producing more melanin in their skin cells. And as UV radiation reaches our cells, our melanin has some ability to absorb that UV radiation to prevent it from ever reaching our DNA. However, melanin can't absorb all of the UV radiation, so some of this still reaches our DNA and can pose a problem and accumulate this damage over time. But what does this damage look like? And UV radiation from the sun can damage our DNA in a couple of ways. The two major ways here are that it can form these bulky dimers where if we have two of the same nucleotide next to each other, the UV radiation can form this connection between them on the same strand of DNA that is not supposed to be there. So now we have this bulky structure that's disrupted the structure of the DNA and prevents other proteins from reading the message that's encoded in the DNA correctly and leading to other problems in the cell. Another type of damage that we might see is that the UV radiation makes it harder for cells to pair the nucleotides correctly across the strands of DNA. So now we have more cases where the cell has made an error in matching one nucleotide to the other across the strand. And as this damage continues to accumulate, we reach this point where the cell now has too much DNA damage to be balanced by its ability to repair the damage. So this damage is going to accumulate in the cell, and these changes to the sequence are going to accumulate. And this is a problem because this unresolved DNA damage can lead to increased risk for cancer. And this can happen through several ways, but one example is that, as I mentioned, the DNA is this coding molecule that includes these important messages in the cells. And some of these messages are going to be for those proteins that are telling cells to stop growing once we have enough of them. And if we accumulate this damage over time, we'll start getting changes in the message of our DNA, and that could lead to a point where the cell is not able to read the message for the stop sign properly anymore, and we lose that signal, causing these cells to accumulate and divide more. And at this point, we reached a place where skin cancer may develop, including melanoma. So kind of back to our original infographic that started us off here, we're started with this fact that sun exposure increases our risk for cancer and specifically skin cancer. And what we've learned walking through this is that this is because UV radiation from the sun causes DNA damage, and this accumulation of damage can lead to cancer. And all of these kind of ways to reduce our exposure to the sun and to UV radiation prevent the UV radiation from reaching our skin. They serve as blocking measures. But I want to take a little bit of time at the end here to go through in a little bit more detail how one of these measures, sunscreen, actually works to protect our DNA from UV. So when we're out in the world, as I mentioned, our skin is very exposed to UV radiation from the sun, especially in the summertime when we're in shorts and short-sleeve shirts. And when we wear sunscreen, it, as the name suggests, serves as a screen or a barrier to this UV radiation. And it can do this in one of two ways. Chemical sunscreens, which have ingredients like avobenzone, actually absorb this UV radiation as they experience it. And then mineral sunscreens, which have ingredients such as zinc oxide, instead reflect that UV radiation away from our skin rather than absorbing it. But both of these lead to the final outcome of a much reduced level of UV radiation actually reaching our skin cells and the DNA inside of them. And certain labels on our sunscreens are actually referring to their ability to protect against UV radiation specifically. One example of this is SPF, for sunscreen protection factor. And rather than referring to, the amount of time that the sunscreen may be effective out in the world, what SPF is referring to is the fraction of UV radiation that's reaching your skin. 1 over that SPF number refers to the percentage of radiation that's reaching your DNA inside your cells. So for example, if you're using a lower SPF, say of 15, this is allowing 1 over 15, or about 7% of UVB radiation to reach your skin. But if you increase to a higher SPF, like SPF 50, now you've dropped that fraction down to 1/50th, or about 2% of the UVB radiation. And the last thing I want to address before we finish up here is another label that is important to look for on sunscreens that refers to this protection is broad spectrum. And if a sunscreen is broad spectrum, it means that sunscreen is able to protect you from both UVA and UVB radiation. So I mentioned earlier that UV radiation is broken into 3 classes, but UVB and UVA are the only 2 that reach us down here on Earth. It was thought historically that UVB, because it's higher energy, is the only type of UV that's capable of damaging our DNA. But it's come to be appreciated over time that even though UVA is of lower energy, it can still cause this damage to accumulate over time. So when sunscreens have this label of broad spectrum, that's important because it's giving you protection against both types of UV radiation that can cause damage to our DNA. So in summary of this first part, we've talked about how DNA is this important instruction manual for telling cells what to do, and that UV radiation from the sun can cause excessive damage to the DNA inside of our cells, which leads to changing the sequence of the DNA in harmful ways. It's important to use measures to reduce the amount of UV radiation that's reaching our skin cells and our DNA, and sunscreen is a really important way to do this and works by absorbing or reflecting those harmful UV rays. So kind of in the overall roadmap of our talk here, we focused in this first part how environmental exposures such as the sun can cause changes to the sequence of our DNA, this first type of nucleic acid here. And then in the second part of our talk, we're going to shift over to Alexandra, who's going to talk about how processes inside the cell to generate these molecules can actually lead to changes or differences in our RNA, the second kind of nucleic acid here. So, thank you.
Yeah, hello everyone. Good evening everyone. Thank you all for coming. My name is Alexandra Bergford. I'm a postdoctoral researcher at the Yale School of Medicine, and it is my great pleasure to talk to you today about RNA. So now you have heard a lot from Kerry about how important it is to maintain an intact DNA, and I'm going to show you today how also variation in RNA can shape health and disease. You have seen this example already. Healthy cells, they divide a few times, and then at some point they just stop growing when there are enough cells or there's no space anymore. However, cancer cells, they just ignore the stop sign and they keep growing and growing and growing, building these tumors. However, our immune system is not completely helpless. There are some instances where the immune system manages to actually kill these cells that are uncontrolled growing and growing. And I'm going to show you today what this has to do with RNA. But first of all, what the heck is RNA? We have all heard a lot about RNA in the news, especially in the past couple of years. And many of us even got injected with RNA in form of a COVID-19 vaccine. But what is RNA? And to address this question, let's first zoom out a little bit and look at the human body as a whole. So we can consider the human body as a network of around 100 trillion cells. And when we look a little closer, then we can see that each of these cells has different compartments, one of which is our nucleus, and the DNA is here in our nucleus. So what is DNA? We have heard about it, and we can imagine DNA as the construction plan that has the information for how to make every single cell of the human body. So now, if every cell has exactly the same DNA, how can it be that a heart cell, for instance, looks different from a skin cell? The answer to that is that all our cells have a different mix of proteins. And I'm talking of proteins here, not only of the protein powder that you may be adding to your breakfast smoothie. I'm talking of proteins here as the elements of the cell. So we can imagine our DNA that has the construction plan for all the houses of an entire city. And then the human body is like the city itself. And each individual cell is like an individual house in that city. And the same way that a skyscraper looks different from a one-family house, also our cells can look different from each other. And every protein then is an element of that house, for instance, a sofa or a door or a window. Now, if we look at our DNA a little bit closer, then we can see that the construction plan for every protein occupies one single page in that book. And if we put our construction plan, like one page, the construction plan for one protein on a scanner, and we connect the scanner to a printer, what do we get? Ta-ta-ta-ta, here's our RNA. So RNA carries the construction plan for individual proteins, while the DNA carries the construction plan for all the proteins. Since this RNA carries the construction plan for individual proteins, we also call it mRNA or messenger RNA because it carries the message for how to make the protein. And to be scientifically correct at this point, I want to mention that there are other forms of RNA floating around in the cell that are not messenger RNA, but today we are only talking about the messenger RNA, the RNA that encodes our proteins. So what is RNA? RNA is a copy of a little piece of the DNA. So now the big question, why do we need RNA? If we have proteins, if we have DNA, why do we need the RNA? And the answer to that is actually quite simple. If we look again at our DNA, and as I mentioned before, it's in the nucleus, and our DNA is stuck in the nucleus. It cannot leave it. However, the site of our protein construction is outside of the nucleus. So how do we get this information now from here to here? And the answer is in our RNA. So we make a copy of only the part of the DNA that we need to make the protein, and this messenger RNA can leave the nucleus and go to the protein construction site. So that's what we need messenger RNA for. And since this may be for some of you a lot of new information, I'm just going to summarize briefly what we have learned so far to bring us all on the same page. We have learned that our body is full of cells that are full of different proteins. We have learned that DNA is in the nucleus and carries the information for how to make the proteins. And we have learned that messenger RNA is a copy of a piece of DNA. It encodes a protein and brings its construction plan to the construction site. So now I have a challenge for you. How can we fit all the construction plans of all the houses of an entire city and all the elements of these houses into a binder that has a limited number of pages? And nature is facing exactly the same challenge. So we have around 20,000 protein coding genes. And with genes, I'm referring here to a unit of the DNA that encodes one protein. So we have around 20,000 pages in this book here. However, there are at least 80,000 proteins in humans, different proteins in humans. And this is just the number of detected proteins. The estimated number is much higher than that. So how do we get from here to here? The answer to that is a process that's called splicing. And I'm going to define to you now what that means. So let's look again at our RNA, at our construction plan that encodes this beautiful sofa here. If we look at it closer, we can see that actually not all of it is encoding this protein here. We have a first part, a coding region that encodes the sofa here, the one piece of the sofa, and we call these coding regions exons. So we have our exon 1 here, then we have a non-coding, and then we have our exon 2 here, which encodes the leg support, and then we have a third exon, which encodes the final piece of the sofa. And since this RNA here still contains non-coding pieces, it's not yet our mature construction plan for the protein, that's why we also call it pre-mRNA. And what's happening then is a process called splicing, where all these non-coding regions here are spliced out. And then, depending on how we recombine our exons, our coding pieces here, we get two different messenger RNAs. One that has this middle piece here, this exon 2 included, and the other one that has it skipped, depending on how we want to recombine here. And accordingly, we can get two different proteins from this one messenger RNA, or from this one pre-mRNA. We can get here a sofa that has the leg support, or one that is lacking the leg support. And that's how we get much more proteins in our cells than we have genes in our genome. And I'm going to show you now an example where this plays a very important role in the body. So let's go back to our cancer cells that ignore the stop sign and keep growing and growing and growing. As I mentioned at the beginning, our immune system is not completely helpless. What's happening is that we have proteins, including the so-called fast protein at the surface of our cells, that can interact with the immune system. And usually, when we have uncontrolled cell growth, the immune system is sending a signal to this fast protein and tells the cells that they should die. So the fast protein is basically causing a self-destruction of the cells. So now, what does this have to do with splicing? Let's look at our fast gene, the gene that encodes the fast protein. We can see here, and this is just depicting some part of the gene, we have several coding and non-coding regions. So here we have exon 5, 6, and 7, the coding regions, and they're interspersed by non-coding regions. So now our pre-mRNA still has the non-coding regions. If we splice now, we can get two different messenger RNAs. One that has exon 6 included and one that has exon 6 skipped. And these two messenger RNAs encode two different proteins. In one case, we have a fast protein that has the middle piece here included. And this middle piece allows the protein to go through the cell membrane. The other, and have one part of the protein sticking out. The other messenger RNA that is lacking exon 6 is building form of this protein that doesn't have the middle piece, so it cannot go through the membrane. It cannot look at the outside of the cell. So while this protein here can interact with the immune system, the other one can't. And accordingly, we have here cell death, and here this protein promotes cell survival, and with this, tumor growth. One other example where splicing plays an important role in the context of health and disease is the context of viral infection. So let's look again at our splicing pattern, where we can have an exon either included or skipped. Now here it is important to notice that while we have one gene that encodes our protein, we usually get several copies of RNA for this gene. So we have, like, for our fast protein now, we have, let's say, 100 copies of RNA floating around in the cell. And not all of these RNAs have the exon here either skipped or included. It's always a ratio by which the exon is included or skipped. So we are interested in knowing what percentage of mRNA of a particular gene has the exon included. And we can visualize this in bar plots. So we can look here, for instance, at 3 different messenger RNAs, 3 different exons, and we always get a percentage by which the exon is either skipped or included here in dark. And now if we add virus to cells, and this is not true for all viruses, but certain viruses, they can cause changes in splicing. And that means that we get different ratios all of a sudden by which the exon is skipped or included. And therefore we get a different protein levels in our cell. Like one protein that was off the cellular machineries, including the splicing machinery. So with this, I hope I was able to convince you that splicing is a very important mechanism that's happening in the cell. And I'm just going to give you a few take-home messages. And these are that messenger RNA is a copy of a piece of DNA that encodes at least one protein. Splicing removes non-coding regions from the pre-mRNA, and variation in splicing determines cell fate. And here my talk ends. So yeah, you have learned about splicing now. And in the next talk, Briana is going to talk about how knowledge of DNA variation can help us to treat Alzheimer's disease. Thank you.
Hi everyone, my name's Briana, and today I'll be talking about Alzheimer's disease and how we can study our DNA to help us better understand it. So the previous talks talked a little bit about how environmental factors can change our DNA, like exposure to the sun. And today I'll be talking more about how the changes in our DNA that we're born with can cause greater association with diseases later in life. So I'll talk a little bit about Alzheimer's disease, the role DNA plays in it, a little bit about my research in the lab, and how we are looking towards a personalized medicine future. So what is Alzheimer's disease? Neurodegenerative diseases affect millions of people in the US, and Alzheimer's alone affects over 6 million people. It's the 6th leading cause of death in the US currently, so this is a pretty prevalent issue in our society, especially with our aging populations. Neurodegenerative diseases are currently irreversible and incurable, and they are mostly characterized by the progressive loss of neuron function. So the brain is made up of many different types of cells, just like our body, and one of the main types of cells are neurons. So the neurons in the brain start to malfunction and they aren't working properly. And one of the main areas in the brain that is affected by these diseases is the temporal lobe, which is associated with memory. So when you think of these diseases, you probably think of memory loss, forgetting faces, forgetting people, not being able to learn new things. So a little bit more, just to stress, Alzheimer's is not a normal part of aging. I think we think when we get older that ultimately we may get one of these diseases, but that shouldn't be the case. This is not normal. And what typically happens are microscopic changes in your brain that happen way before the first signs of memory loss. So right here I have a picture that's showing a patient with Alzheimer's disease versus a normal brain. And what you can actually see here is that with this disease, your brain actually starts to shrink. And that's because, like I mentioned before, the neurons actually, the cells in your brain start to die off. So your brain actually starts to get smaller. Like I mentioned, we are made of many cells, and the brain is made up of neurons and glial cells. So neurons are kind of like the messengers of the brain. They're sending signals to each other, and they're sending signals to other parts of the body. There's the dendrites, which help neurons talk to one another. There's the cell body that contains the cell nucleus, and that's where all of our DNA, our genetic information, is stored. And then there's the axon, and that's what conducts these electrical impulses. So this is what I'm showing here. Really cool. You can actually physically see the electric signals from these neurons. So for instance, if I wanted to raise my left hand, my neuron in my brain would have just sent an electrical signal down to my arm to make me raise my hand up. Another type of cell in the brain are glial cells. And these are kind of like the maintenance workers of the cells. They're making sure that everything's running smoothly. If any debris or anything bad's in the brain, they're going to start to go to that area and clear it up. And one of the most important cells in these diseases, like Alzheimer's, is microglial cells. And this is something that I'll talk about a little bit later. But these are the innate immune system of the brain. They are the ones that are going to like debris in your brain and making sure that they're trying to clear it up. Let's just talk a little bit about DNA and their role in Alzheimer's disease. So like we mentioned, we're made up of cells, and inside cells are our DNA. The DNA is actually pretty long. Inside a single cell, it's around 2 meters. So to get it all into this little nucleus, the DNA has to be wound around a chromosome. So the chromosome you can kind of think of as a ball of yarn, is a ball of our DNA. So our DNA contains all these nucleotides, And so variations in these nucleotides can increase our risk of getting diseases later in life. So one person may have a C nucleotide here, and another person may have a T. And so what we study in the lab is how these small little variations in our DNA can increase our risk of getting disease later in life. So to do this, one of the main ways we can do this is looking at graphs. And this is called a Manhattan plot, and it gets its name because it kind of looks like the city skyline of New York City. So what we're looking at here is a chromosome on the x-axis, so all of our chromosomes, and then on the y-axis is the association with disease. So all of these little dots are different genes in our genome. When we're looking at these plots, we want to look at the dots that are higher up on the graph because they're more highly correlated with, say, neurodegenerative diseases. So you can get a graph of over 10,000 people's genomes, and we can kind of study the patterns that are happening in people with these diseases. So if I was looking at this graph, I'd want to pick out the genes that are up here first to study because they have the most association with disease. So in my lab, we use stem cells. So stem cells are a pretty cool type of cells. They have the ability to become pretty much any type of cell in your body. So in my lab specifically, we can use stem cells from people's skin. So we can take a person's skin and transform them into stem cells. And with different foods, we can create blood cells, bone cells, and in my lab, we're interested in nerve cells. When we now we can make the cells of the brain. So what's been stopping us in the past is that we can't just take a part of somebody's brain and study it. So it's been pretty hard to study these diseases because we've only been able to analyze like post-mortem samples, so samples from people that have passed. So now with the advent of stem cells, we're able to sort of mimic what's happening in real life and be able to study these cells while they're still living. So what we do is we can take these stem cells and just with different foods, we can push them into different fates. And what I mean by that is we can create blood stem cells, and then from here we can make microglia. And we do this because during development, our blood stem cells go up to the brain and create the microglia, which are the immune cells of the brain. So this mimics closely to what's happening in development. And then here I have like a nice picture of some stained microglia under a microscope. And we just do this to make sure that the microglia are expressing genes that are known to be expressed in human microglia. So now we can have some pictures that I've taken in my lab, which I think are kind of cool. So these are the original stem cells. They kind of create these colonies that are touching because they always want to be in close communication. I give them stem cell food, and then when I'm ready to start the microglia process, I give them different food, and we can create these blood stem cells. So what's different from these two types of cells is these are very circular cells. They're not sort of clumping, they're kind of on their own at this point. And then with different food, I can create microglia, which is really awesome. And what's really cool about microglia is that they're very different looking. Here we have sort of uniform colonies and uniform circular cells. Microglia are kind of doing their own thing. And that's because, like I mentioned, they're the immune cells. So they're constantly reaching out in their environment. They're surveying the area to make sure nothing bad's happening in the brain. They're all talking to each other because they want to say, like, hey, if there's something going on over here, we all need to go and help fix it. So how does all this fit into how we can treat Alzheimer's? So in the future, I think what we're going to see is sort of a personalized medicine approach. And so typically when we think of treatment right now, we think of a one-size-fits-all where we make one drug and we hope that it works for everyone, but we know that that's not the case. Sometimes there's serious negative effects from drugs. They don't work for everybody, and so then that leaves people without any options. So now what we can do is we can analyze everybody's genetic makeup, what our DNA is made of, and we can analyze that individually and hope to make personalized medicine tailored to that specific person. If one person had variant X, then they would be given this drug, and somebody else may be given that drug. And what we hope for is that these have better results, that they're tailored to the person's genetic makeup, and that they won't have serious negative effects. So I think this is a really cool future ahead of us. And so the key takeaways for all of our talk is that nucleotides are the building blocks of life, and changes in them can cause disease. Studying DNA variants and variants in RNA can help identify individuals at higher risk for diseases such as Alzheimer's or cancer, and can help us in aiding early intervention and prevention strategies. And understanding these variants can help us create tailored treatments tailored to an individual's genetic profile, potentially revolutionizing Alzheimer's and other types of diseases. So with that, I'd just like to say thanks.